Hello everybody. We're going to talk a little bit more about genetics and we're just going to head right in. Alright, so to refresh our memory, each characteristic that you have is inherited by your parents. And what inheritance means is that they are the genetic characters that are transmitted from parent to offspring collectively. These characteristics that you inherit are called traits. An example of a trait would be like your eye color, your hair color, your height, etc. It's basically what makes us unique from each other. A gene is a basic unit of heredity. It is a sequence of nucleotides that are in DNA. So, backtracking that a little bit, the nucleotides that are in DNA are the adenine, thymine, cytosine, and guanine. And we also learned about someone. His name is Gregor Mendel. He's an Austrian monk who discovered the science of genetics using pea plants. So more about Mendel. He made this law named the Law of Segregation. And the Law of Segregation is the process in which genes are transmitted from parent to offspring through reproduction. He studied this with pea plants, their pod color, seed, etc. And the gene that we're going to concentrate on for this lesson is just pod color, so you don't have to worry about the seeds or anything for right now. So we're just going to concentrate on pod color. Some pea plants have yellow pods and others have green. Pea plants can self-pollinate, which means that they can reproduce using their own DNA. So Mendel had a true breeding yellow pod and a true breeding green pod, pea plants. Now, what is true breeding? True breeding is when a gene can only reproduce that specific gene, and so there are no variables. So, example, a true breeding yellow pod can only reproduce or produce a yellow pod offspring, and same thing goes for green or any other trait. But for this instance, let's just say if a green pod is a true breeding green pod, then it would only produce a green pod. There's no other way that it can produce anything else but a green pod. Now, Mendel knew that a yellow pea plant will produce a yellow offspring. So what he did was he cross-fertilized them with a green pea plant. So he didn't let them self-pollinate themselves. He crossed both a yellow pea plant with a green pea plant. And the true breeding yellow pea plant and the true breeding green pea plant were named the parental or P generation. Now, the product of that cross-fertilization was called the first filal or F1 generation. Now, as you can see here, you have a true breeding green pea plant with the green pods, and then he crossed them with a true breeding yellow pod. And what he got was all these 4 offsprings, which were the F1 generation, and he got them all green. The result of these 2 being crossed was all green pods. Now, once he found out that the offspring came out green, he let those plants self-pollinate themselves, and the result of those unions are called the F2 generation. So it's basically like the children's children, as you can say. So the F2 generation consisted of 3/4 green and 1/4 yellow. Now, how did that yellow gene appear? So if you see here, the F1 generation green pod self-pollinated with an F1 green pod. You got 3 greens and 1 yellow. Now, you may think green and green, you would get all green, but this wasn't the case here. Now, because of this discovery, he made the law of segregation. Along with this law are 4 points. So here they are. He determines that genes can come in different forms. So the gene for a green pod color is represented by an uppercase G, and the gene for a yellow pod color is represented by a lowercase g. And there are two alternate forms of the same gene. They are called alleles. So right now we know that alleles are two alternate forms of the same gene. And the reason why there are two alternate forms is because one allele or one form comes from our father and another comes from our mother. So he figures out that the alleles that they separate or segregate during gamete formation, and they come together during reproduction at random. So your one of your parents has one set of DNA. So in, let's say for pod color, your father has one set of a gene, and your mother would have a different set of a gene. So when the pair of alleles are different, one is dominant and the other is recessive. Now, what's dominant and what's recessive? Now, dominant just means that it's a trait that is expressed. It is represented by a uppercase letter. So, for example, in the green pod color is dominant because it can be seen. It is the most aggressive form of this gene. Now, a recessive is the trait that isn't shown. It is represented by a lowercase letter. Example, the yellow pod color is recessive because it isn't seen because the green overpowers it. But the gene is still, the gene for the yellow pod is still there. So let's take a look at the next slide and we'll see about with these alleles. So here you go. Now, remember, this is from the F1 generation. This is the P generation and P generation. He crosses the true breeding green pod with the true breeding yellow pod. So remember, the green is dominant, so he has them named with the capital G and capital G. And remember, it's two alleles because one comes from a mother and the other one comes from a father. And then the same thing goes with the yellow. There's a lowercase g here and there's a lowercase g here. And then this is the cross. And this is how it comes about. Like, this is why the F1 generation all came out green. Now, we have two sets of alleles to represent the same gene. In this case, we will use the alleles capital G and lowercase g for pod color, just like before. So to properly call a yellow pod color gene, we would write lowercase g instead of just one lowercase g. Each G represents one of our parents. Now, there are two different ways to categorize these alleles. Now you have homozygous. Homozygous means two of the same alleles. Just remember the prefix for homo means same. Now, for example, uppercase G and uppercase G, this example right here, can be called homozygous dominant because they are both capital letters. They're uppercase letters. And you have a lowercase g can be called homozygous recessive. Heterozygous. Heterozygous are different alleles of the same gene. So hetero is, the prefix hetero means the same. It means different alleles. And this is represented this way. You have capital G and lowercase g. It is called heterozygous. And because the dominant gene is present here, the color of the pod would have to be green, since green is the dominant color. Genotypes and phenotypes. So genotype is basically the genetic makeup of the gene. So what we were just talking about before, homozygous dominant, homozygous recessive, and heterozygous, they are the genotypes for, in this example, for the pod color. Or so when you call something like homozygous dominant, or homozygous recessive, or heterozygous, you are basically letting them know the genotype of a specific gene. And phenotype is the visual expression of the gene. So if the genotype is either homozygous dominant or heterozygous, its phenotype would be green in the case of the pod color. If the genotype is homozygous recessive, its phenotype would be yellow. So it's basically what we would see. Would be our phenotype. So a Punnett Square. Reginald Punnett invented a way to figure out the probability of a gene appearing in an offspring. So in the 9th slide, there's an example of a Punnett Square. So let's just go back to that slide. All right, so here you go. This exactly is called a Punnett Square. So here you go. And what it is, it's one large square that is divided into 4 different squares. Okay? And what they did here, what Reginald Punnett did, was he separated each allele. So you have here 1G goes into this one mini square, and you got this other G going to this other mini square. And you got this G, this capital G, going into this side right here, and this G going into this side right here. So what he does is he crosses them. Okay, so then he crosses this G with this G, becomes this G. This G and this G becomes this G. And this G crosses over from this one and becomes this. And this gene goes with this gene, and here you go. So let's just go up ahead here. So he has separated the alleles into a different mini square, and he crosses each mini square so you can find out the probability of specific alleles. So going back to slide 9, we saw that there is a 4 out of 4 chance, or 100% chance, that the offspring would be heterozygous. So let's just see about future generations. Let's just have, I already have a problem set up for you guys. Let's see if we can do this. All right, so if a pea plant that was heterozygous for pod color fertilized with a homozygous recessive pea plant, what is the probability of their offspring being heterozygous? List the possible genotypes and phenotypes. So to refresh our memory here, heterozygous is basically the capital G and the lowercase g, and homozygous recessive is the lowercase g and lowercase g. So using the Punnett square, we made our square here, or looks like a rectangle, but it still works. So then we have our Punnett square here, and then you would just divide it, these two, straight ahead here, and then just cross them. So we got this G crosses with this G, makes this G. This G and this G makes this G. And this G and this G makes this G right here. And this G and this G makes this G. So the question was, what is the probability of the offspring being heterozygous? So I have it here in red for you guys, right here. The probability of the offspring being heterozygous is 2/4 or 50%, because there are 2 out of the 4 are heterozygous. And the possible genotypes, you list them at whichever, like your whatever in the boxes here. So you got a heterozygous here and a homozygous recessive here. So that's your possible genotypes for a future offspring, and your possible phenotypes. Because you have heterozygous, the possible phenotype would be green. And since you also have a homozygous recessive, another possibility for the phenotype would be yellow. All right, so your assignment would be I have made a worksheet for you guys that I have emailed it to each of you, and that you would bring in next in class. And here are my credits, and there you go. If you have any questions, you can always go back to this slide, or you can always email me. Thank you. Bye.